those moments that really stick with you? For me, one of them was learning about ATP synthase. So, this amazing molecular motor inside of your cells that makes ATP. I remember so vividly being introduced to it in my undergrad biochem class, so I'm so psyched to be able to tell you all about it, as well as basically how it gets its power to make ATP from the electron transport chain.
Looking at the bigger picture, we started by talking about glycolysis, so breaking down sugars. And glycolysis, well, that got us to pyruvate. We want to take pyruvate and oxidize it further. In order to do this, we first had to convert it to acetyl-CoA, and then that could enter the citric acid cycle. And now we're after the citric acid cycle. What are we going to do? Well, we've created some ATP already, but the real big payout is going to come from this NADH and this FADH2 that we've created along the way. If we want to cash in that for ATP, well, now we're going to use oxidative phosphorylation. We're going to go through the electron transport chain and make ATP through this oxidative phosphorylation process. So, oxidation and reduction. Good old oil rig. Oxidation is loss of electrons and reduction is gain of electrons. If we pass electrons from something with a lower reduction potential, so something that doesn't want the electrons as much, to something with a higher reduction potential, something that wants the electrons more, that's going to be energetically favorable. So this is going to release energy. So if you hear energetically favorable, think energy is going to be released. So we want to transfer electrons from a lower reduction potential to a higher reduction potential, because remember, reduction, that oil rig, reduction is gain of electrons. If we pass electrons to something that wants it more, we're going to make the molecules happier. And if we make molecules happier, we get energy. We can then use this energy to drive processes like the production of ATP. But we need to controllably release this energy if we want to do that. If we look at, say, that NADH that we generated during the process of glycolysis and during the process of our citric acid cycle, our TCA, our Krebs cycle, whatever you want to call it, that NAD has a lower reduction potential than oxygen, much lower. This means that if we were to pass electrons directly from our NADH to oxygen, we'd get a big boost of energy. But we don't want just big booms going off in our cells. Instead, we want to controllably release that energy in a series of little steps. We can couple that energy to things like the movement of protons. And why would we want to do this? Well, in our mitochondria, we've got this situation where we have these different compartments. We can pump protons out of the inner part of the mitochondria, the matrix, into this intermembrane space. We can then build up a proton gradient, and not only is this going to be a chemical gradient, so because we have these physical entities, these different chemicals, we have this ion gradient, but we also, because these are charged, we're also going to have an electric gradient. So we're going to have an electrochemical gradient. We're going to be building up all of these protons in this inner membrane space. And then what's going to happen is those protons are going to flow through this ATP synthase like a dam that uses hydraulic energy. But in this case, we're using this proton motive force. Basically, we create this gradient that we're going to then use to drive the flow of the electrons through this pump that is then going to cause the pump to move, and it's going to cause ATP to be made and released, and then this can keep going and going. So we're coupling the passage of electrons to the passage of protons, which ultimately is going to drive the production of ATP. That's the big picture. By transferring these electrons in these little steps from one carrier to the next carrier, we're going to be pumping electrons because we're using the energy from those transfers from something with a lower reduction potential to something with a higher reduction potential. Energy released, energy used to create that gradient. That gradient is then going to drive the production of ATP. And now we just need to talk about all the nitty-gritty details of how this is all going to go down. First of all, let's talk about where this is all going down. When we talked about glycolysis, that was happening in our cytoplasm. When we talked about the citric acid cycle, and when we talk about the oxidative phosphorylation and our electron transport chain and all that stuff, now we're going to be dealing with our mitochondria. So remember that our mitochondria, they came from basically an early cell swallowing a bacteria or similar prokaryote, and then therefore they have this kind of two membranes. So you've got this room within the room within your cell. This inner mitochondrial membrane is going to surround the inner room, this mitochondrial matrix, which is where our citric acid cycle was happening. The stuff that we're talking about today is mostly going to be taking place in this inner mitochondrial membrane, as well as in some of it in the matrix and some of it in this outer room, this intermembrane space. So you've got this outer mitochondrial membrane, you've got this inner mitochondrial membrane. Between them, you have this inner membrane space. We're going to be pumping protons out of the matrix into this intermembrane space. They'll then flow back into the matrix and generate the ATP on the matrix side, which can then get like swapped out so that it can flow out. This outer membrane is going to be much more permeable to things than this inner membrane. So this is going to make it so that we're going to be able to have things move more freely into this inner membrane space. But if you want things to go inside, well, now you're going to have special transporters, and this is going to come into play throughout. A quick note about mitochondria. So I've drawn them as kind of that simple cartoon, but mitochondria are far from simple. And they're not always a bean shape. So that was how people first saw it under a microscope. It looked like a bean, but it actually, mitochondria can look very different in different types of cells. Cells can also have different numbers of mitochondria. So our muscle cells, say, will have tons and tons and tons of mitochondria, whereas other cells that don't need as much energy won't have as many. Mitochondria can also do things like fuse together. And within a single mitochondria, you can have different amounts of kind of the folding of that inner membrane to give you these cristae. The more folding you have, kind of the more surface area you have to have those electron transport chain components embedded in them, so you can make more energy. In order to make all of those proteins that make up the respiratory chain, there you're going to have to make some of the proteins actually in the mitochondria themselves, and some of them are going to get brought in. So it's all this complicated stuff, but your mitochondria actually have their own genome, and it has their own ribosomes, and they actually make some of the proteins of the electron transport chain. We're not going to go into the details, but it is pretty cool. Going back to our big story, though, let's talk about how we're going to use the mitochondria and use that compartmentalization as a way to power the production of ATP. This is there's this chemiosmotic model. This is going to describe the coupling of ATP synthesis to the electrochemical proton gradient. So that's that gradient we were talking about before. We've got the chemical gradient, where we have basically the difference in proton concentrations is the difference in the concentration of a chemical. So we've got a chemical gradient. And we have an electrochemical gradient because we have these protons are positively charged, and if we separate them, well, then it's like a battery. So we're getting this electrical gradient. Put them together and we get an electrochemical gradient. When you move things down a gradient, that is going to be thermodynamically favorable. That's going to release energy, and that's going to be the energy that we're going to be using. So this energy that we create through the pumping of the protons is going to be used to generate our ATP. But we need to do that, create that gradient, and that gradient is going to be created through a series of redox handoffs. Remember that we want to move something with a lower reduction potential to something with a higher reduction potential. And therefore, we're going to need to have different molecules that can give and take electrons. And we want a variety of different molecules with different reduction potentials so that we can do things stepwise. And so that something will kind of always want it more until ultimately we get to oxygen, which wants it so bad. We saw a couple electron carriers when we talked about glycolysis and we talked about our citric acid cycle. These were going to be FAD and NAD. And then we also discussed how we could have like NADP plus, which was going to be used primarily for anabolic things. But our FAD, so our flavin adenine dinucleotide, and our NAD, our nicotinamide adenine dinucleotide, we saw that in glycolysis, we created the reduced form of, we reduced NAD plus to give us NADH. And then in our citric acid cycle, we reduced FAD to give us FADH2. Keep this one in mind because it's going to come into play as actually part of our, that's going to be our kind of, the step where we create that is actually complex 2 of our electron transport chain. So stay tuned. But first of all, let's think about some of the similarities and differences between NAD and FAD. I know that I'm going to be kind of lazy about saying like NAD plus or NAD. Just go with it. What's the difference between them? Well, FAD, this is actually going to be tightly held in proteins. We call those flavoproteins. And whereas our NAD, this is going to be soluble, so it can go and travel around. The FAD is typically going to have a higher but variable reduction potential than our NAD. If it has a higher reduction potential, then basically, if we were to pass something to it, we would get more energy. And so we can use it to drive oxidation reactions that require more energy. Things like, say, making a double bond from 2 carbon single bonds. Whereas with the energy needed to, say, go from an alcohol to an aldehyde, for that, we could use NAD. But for going the harder reactions, we needed to use FAD. But if we use FAD, well, now we're at a higher reduction potential. And so we'll get less gain when we pass it off to the next thing in the line. The FAD reduction potential, though, is going to be variable because it's tightly held in the protein, and that's going to, different proteins will have different environments that are going to change the actual reduction potential. But it's typically going to be higher than your NAD. But those are far from the only electron carriers, and we're going to see a bunch of different electron carriers when we talk about the electron transport chain in this video. There are 5 main types of electron carrying molecules. So we talked about our NAD or NADP. We also have our flavoproteins. And so we talked about FAD, but there's also this a similar molecule, FMN, flavin mononucleotide, as opposed to flavin adenine dinucleotide. So very similar, but also a flavoprotein, so also very tightly held. And that is a molecule that is going to come into play in complex 1, as we'll see. Ubiquinone. So this is also known as coenzyme Q or Q. And this can move through the membrane. And then we have cytochromes, which are proteins with iron-containing heme groups, as well as iron-sulfur proteins. And so we'll talk a little bit more about these different ones. So remember when we talked about NAD and we talked about FAD, those were transporting pairs of electrons. Those were doing hydride transfers, remember? So we had that pair of electrons that was moving. When we talk about some of these metal carriers, when we talk about iron, when we talk about copper, these are going to take 1 electron at a time. And so we need a molecule that can go from 2 electrons, take 2 electrons and give 1 electron and that sort of thing. And it turns out that coenzyme Q is really great for this. So coenzyme Q is basically, it's got this little group where the action is going to be taking place, but then it also has this long hydrophobic tail. This long hydrophobic tail is going to allow it to hang out and move around the membrane. So that's going to be important in the CoQ's role of transporting electrons from one place to the other, that it's able to move through the membrane. You might have heard of like Q10 supplements. Basically, that 10 refers to there being 10 carbons in this chain, this R chain. But we're not going to focus on that chain right now. We're going to focus on where the actual oxidation and reduction are taking place. In ubiquinone, so our Q, This is going to be the fully oxidized form. Then if it takes an electron and a proton, well, now we get this stabilized free radical. So remember, a free radical is when you have a lone electron. So not a lone pair, but an actual lone electron, unpaired. This is going to be our semiquinone radical, or our UB semiquinone. And so you'll see it drawn like this. What then happens is if it accepts another electron and proton, well, now you're going to get the fully reduced form, which we call ubiquinol, or QH2. So this is the fully reduced form. It required 2 electrons and 2 protons. And therefore, it's going to be able to give and take 2 electrons and 2 protons. And we can use it to both transfer protons as well as to transport electrons. It's ubiquinone, the name kind of suggests it's ubiquitous. It's going to act as a sort of universal carrier because of its role of being able to accept 1 or 2 electrons, and it's going to be able to take electrons from both our NADH as well as our FADH2 or our FMN. What about iron? So iron, we can have that like +3 state, that +2 state. Basically, it can give and take electrons, but it needs an escort. We need to keep it safe because we don't want things giving and taking electrons willy-nilly, or else we'll get unwanted radicals. So radical in the case of our ubiquinone was helpful because it's just this transitory thing. We'll see, however, how it can actually, if it escapes, it can cause oxidative stress, oxidative damage. When you have free radicals, those are often very reactive. We don't want metals just taking electrons or given electrons in an uncontrolled manner. And therefore, when we use atoms that, when we use metal ions that do give and take electrons a lot, things like iron, things like copper, we're going to use them in a tightly controlled manner where we have them held very tightly in things like proteins. And even within proteins, we'll have them held tightly, such as in prosthetic groups. So these tightly held molecules like heme groups. We saw heme in the case of hemoglobin, but it's also going to take to be here because, well, it's really good for holding onto iron. Also good for holding onto iron are like cysteines. So the sulfur, so this would be like an iron sulfur cluster, as well as you can have them coordinated in these things called like Reski proteins, where you have them coordinated by histidines. So various ways we can hold on to those metals and take advantage of their ability to give and take electrons. So we'll see these different molecules be used, things like our cytochromes, so our proteins that hold on to one of those heme groups and that holds on to the iron. We'll see our ubiquinone. We'll see oxygen ultimately, and we will see this NAD and the FAD. All of those are going to come into play in our electron transport chain. Now we need to look in a little more detail about how this chain is going to work. I think it's best to start with kind of getting an overview of what it all actually looks like. So there's this great animation from the Harvard X consortium thingamabob. And so why don't you go watch that, and then we'll discuss in detail. All right, so we've got these 4 complexes, and really complex 2, we can take that out and it wouldn't matter. It would matter in the terms of our final yield. But complex 2 is really just an alternative way to give electrons to Q. So remember Q, that's our ubiquinone. That's going to be the transferring, we can give electrons to it from things like NADH, which we do in this complex 1, or from things like FAD, or which we get indirectly from succinate, which is complex 2. There are also other ways we can get things to Q, and we'll discuss that later. But the role of complex 1 is to get those electrons to Q. The role of complex 3 is then to take those electrons from Q and pass them on to cytochrome C, this little protein with one of those heme molecules that's holding onto the iron that can hold onto the electrons. And then the cytochrome C is going to go, and it's going to go to complex 4, which is going to pass those electrons on to oxygen. And then this is all in these complex 1, complex 3, and complex 4, here you're going to be pumping out protons, and the pumping out of protons, then we'll talk about how we can create ATP. But first, let's look in detail about how each of these complexes work. Don't get tied down by the nitty-gritty details, but take a look but start thinking about kind of the logic of the pathway and how we're passing electrons off. So in the case of complex 1, this is also called NADH ubiquinone oxidoreductase or NADH dehydrogenase. We're removing electrons from NADH, so we're oxidizing it, and we're reducing ubiquinone. So we're passing those electrons onto ubiquinone. But we're not doing that pass off directly. Instead, we're going to do it through a series of electron transporters. This starts with FMN. So remember, that was one of those. This is then like a flavoprotein. So FMN was our flavin mononucleotide. It was like our FAD, but slightly different. And it's going to be held in this complex 1 protein. It's then the electrons are going to be transferred from that through a series of our iron sulfur centers, ultimately to our coenzyme Q. Each of these pass-offs is going to be energetically favorable because we'll be going from something with a lower standard reduction potential to something with a higher standard reduction potential. We're going to couple the energy from those favorable transfers to the pumping of protons. So for each NADH that goes in, you're going to get 4 protons pumped in complex 1. And you're going to be reducing 1 Q molecule, giving it 2 electrons. So you're fully reducing it to its ubiquinol form. So we went from ubiquinone to ubiquinol. We now have this QH2, this ubiquinol, that we can use to pass energy on further. But first, before we go back to our story about the ubiquinol, we need to talk about another way we can form ubiquinol. And that's going to be our complex 2. So our complex 2, well, this just passes electrons to Q. And so this is succinate dehydrogenase. Yes, it's that succinate dehydrogenase. So it's that one from the tricarboxylic acid cycle, or Krebs cycle, or citric acid cycle. It was the step when we went from succinate to fumarate, and we actually made that FADH2. When we made the FADH2, this was actually happening in that inner mitochondrial membrane. So most of our citric acid cycle was happening inside of the matrix. But this step was actually happening in this inner mitochondrial membrane. This way, we have the electrons directly passed into the electron transport chain at this step. This is important because that FAD, remember, that's one of our flavins, that's going to be tightly held in a protein. So if we want to use this as an electron transfer, we need the molecule that's holding it, the enzyme that's holding it, to actually be where we need it. And so by having it embedded in this inner membrane, it's able then to directly transfer those electrons from succinate onto FAD and then onto ubiquinone, so our Q. So it's going to pass the electrons from succinate to FAD, then ultimately, through some iron sulfur centers to ubiquinone to give us ubiquinol. What's a little confusing is, yes, we're going to be transferring these electrons, but we're not actually going to be pumping any protons. So you might wonder, like, well, what's the point? Well, the point is to get the molecules to ubiquinone. So our ubiquitous carrier, that thing that's going to be able to transfer those electrons further down the line, we need the electrons to be there. So we're going to waste the energy from this in order to do that. Waste in the terms of we're not pumping out protons, but we're still doing something very useful. Why aren't we pumping out protons? Well, if we think back to the energy of our FAD versus the FADH2 versus the NADH, remember that our FAD had this higher reduction potential, which means that it's going to have less of a difference in reduction potential between it and Q than there would be between NAD and Q. And therefore, we're not going to get as much energy from this pass off, but we still are going to get that ubiquinol. Now that we have that ubiquinol, let's go and let's take it further. Let's use it to transfer those electrons to cytochrome C. So cytochrome C, remember, that was going to be kind of like our relay protein. It's going to be this small little protein that can move around this inner membrane space. And it's going to be able to take those electrons from ubiquinol and transfer them to the fourth complex, which is where we're going to be transferring them to the oxygen. So complex 3 is going to also be called cytochrome BC1 complex, or ubiquinone cytochrome C oxidoreductase. So these long names, complex 3, a lot simpler to say. And it's going to basically pass electrons one at a time from our ubiquinol to cytochrome C. So remember, ubiquinol is fully reduced. It's got those 2 electrons it can transfer. Cytochrome C, it's going to only transport 1 electron. So cytochrome C, it's got that heme group that has the iron, and the iron is only going to be giving and taking 1 electron at a time. So we're going to need to just give cytochrome C 1 electron at a time. But we have 2 electrons in our Q, in our ubiquinol. And so we don't want to waste that second electron. Instead, we're going to go through some complex gymnastics in order to take ubiquinol and pass one of the electrons from it to the cytochrome C and kind of hold on to that other electron until we go to a second cycle. So there's this thing called the Q cycle. I don't need you to worry about the details, but I am just going to explain it so you have an idea about what's going on. Basically, you have these 2 binding sites for Q. You have this QH2, so our ubiquinol enters, and our ubiquinone enters, so our Q. So we have one that's going to be in this fully reduced state and one that's going to be in this fully oxidized state. What happens is that fully reduced one can now pass off those electrons. So it's going to pass 1 electron to cytochrome C. It doesn't pass it directly to cytochrome C. Instead, it goes through an iron sulfur center, but then it'll go to cytochrome C, and then cytochrome C can then move away and go take it off to complex 4. But we still have that second electron. That second electron, it's going to go through a series of heme molecule carriers, and it's going to actually reduce that ubiquinone that we had down here. So remember, we had ubiquinol up here, we had ubiquinone down here. The electron from ubiquinol, one of the electrons, kind of like the extra one that we couldn't directly pass to cytochrome C because cytochrome C would only take 1. We're going to take that electron and we're going to give it to this ubiquinone. This is going to generate that free radical, that semiquinone radical. Now what's going to happen is that this is going to leave us with another oxidized Q, and that Q can then move out and it can go and it can re-enter the pool. In the process of doing this transfer, we're also going to be transferring those 2 protons. So we're pumping out 2 protons in the first cycle. At the end of the first cycle, basically what's happened is that this free radical that we've generated, it actually moves back into position on this site. In order to reduce it fully, we're going to need to add 2 protons, and we're going to need to add another electron. We can give it another electron if we take another ubiquinol. So we take another of the fully reduced forms in. We take an electron from that and give it to this free radical to create QH2, to create our fully reduced ubiquinol. The second QH2, well, now it's going to give the other electron to cytochrome C. And so in this process, we basically passed off another electron to cytochrome C. And in the process of doing this, we're going to give off another 2 protons. And so we have the same cycle happening up here. But down here, this is kind of where we're going between having we're going from the fully oxidized to the semiquinone. and then to the fully reduced, our quinol, so our ubiquinol. So bottom line is we're going to be passing those 2 electrons from QH2 onto 2 cytochrome Cs and pumping out 4 protons. But it's not going to be as simple as just doing it straight like that. We're going to do it in a couple of cycles, but it is going to happen. Now you've got those cytochrome Cs that have those electrons. So at complex 4, now we're going to go and we're going to take those electrons from cytochrome C and pass them on to oxygen. Oxygen wants them really, really badly, remember? And so this is going to be highly energetically favorable. We're going to use it to drive the pumping of protons again, because we get that big boost, we get that protons pumped. And in the process, we're going to be making water. The numbers get a little weird here because when we think about reducing oxygen, well, oxygen, molecular oxygen hangs out as O2. And so we're actually going to take O2 and we're going to add 4 electrons to it. So we're going to need 4 cytochrome C molecules in order to do this. And we're going to create 2 H2Os. In the process, we're pumping out 4 protons. But if we were to think about it from the perspective of that initial NADH that was going into the line or something like that, well, then it would be the equivalent of 2 protons. So 2 protons per oxygen, but because it's molecular oxygen, it's O2, then it's going to be 4 protons overall. How this transfer is happening is it's going to be happening through several carriers. These include cytochromes A and A3, as well as copper ions. So you've got this kind of like iron and copper working together. So complicated transfers, but ultimately you're basically the same story over and over. We're passing electrons from something with a lower reduction potential to something with a higher reduction potential, generating energy, using that energy in this case to pump out protons and boost that proton gradient. Putting it all together, we can think of these 4 complexes. We have complex 1. We're going to take electrons from NADH, pass them off to Q. This is going to take our ubiquinone and give us ubiquinol in our Q pool. We can also give electrons to Q through succinate dehydrogenase, or complex 2, which is the same succinate dehydrogenase that we had in that electron transport in our citric acid cycle. This is another way we can give electrons to Q. And it turns out that there are actually a lot of different ways to pass electrons to Q, including from things like fatty acid oxidation. So in fatty acid oxidation, we're going to need to do one of those reactions that requires the formation of a double bond and between 2 carbons. So we're going to use our FAD. And this is actually going to then contribute directly those electrons into this Q pool. So we talk about this kind of like general Q pool, because it doesn't really matter where those electrons came from that are given to Q. Now you've got that reduced Q, you've got your ubiquinol. So wherever the source of the ubiquinol, now it can come and it can enter this complex 3. In the complex 3, those electrons from ubiquinol are going to be passed to cytochrome C. Cytochrome C, this is this little protein that can move in this inner membrane space. It's going to go and travel to our complex 4, which is where those electrons are then going to be passed off to oxygen, giving us water. In complexes 1, complex 3, and complex 4, where we get enough of an energy boost from those electron pass-offs in order to pump protons out. But with complex 2, here we don't. There's not as much of an energy difference in passing those electrons from the FAD to Q than from NADH to Q. We don't have enough energy here in order to drive the pumping of protons, but we are able to contribute to this Q pool that we can then use to pump protons down the line. So if we think about electrons entering from NADH, well, for each NADH, we'll get to pump out 4 here. We'll pump out 4 here, and we'll pump out 2 here. And so that would be the equivalent of 10 protons pumped out per NADH. And remember here, this is looking at it in terms of a single oxygen, so half an O2. So that's why you see this 2 protons as opposed to 4 protons pumped out, because we're talking about in terms of this kind of like half of the reaction happening. In the case of if we were to start with our FAD, if we were to start just by adding something to Q through one of these ways that doesn't directly go through complex 1, well, now we're only going to have these 6 protons produced, these 6 protons pumped out. So when we come to accounting later on, we'll have to keep that in mind, that we're getting more energy. We're getting more protons pumped out if we enter at complex 1, but we can also enter at complex 2 or any other route to Q. And although we kind of look at these as these isolated things, actually these complexes often hang out in these kind of bigger groups called respirasomes. So the complexes themselves are often very complex and they have lots of different proteins. And then they hang out together in these respirasomes where they're kind of close together in the membrane, which makes sense because, well, you don't want those electrons flowing all over the place, out of control. Speaking of out of control, in the electron transport chain, remember that we're ultimately passing off electrons to oxygen. What would happen if we didn't have oxygen around? Well, then there wouldn't be anything to accept, or we didn't have enough oxygen around. There wouldn't be anything to accept the electrons at the end. We'd just have a slowdown in our electron transport chain. If we have a slowdown in our electron transport chain, this can lead to the generation of reactive oxygen species, or ROS. So remember how Q, it could accept one electron, it can accept 1 electron or 2 electrons. And if it accepted 1 electron, we got this radical. And this radical was going to be generated in kind of like the normal process of things. But in the normal process of things, that Q was going to be kind of going straight from one thing to the next in that inside of the complex, and it wasn't going to be directly able to harm anything. It wouldn't be near any oxygen. All of that would be happening in this very controlled manner. But if there are holdups, it can kind of like escape. When you have that radical escape, well, then it can go and it can do things like cause damage. In order to prevent it from causing damage, our cells have different enzymes that can kind of counteract the oxidative stress. So we've got this like superoxide dismutase. We've got glutathione peroxidase. We have ways to take care of it. And it turns out that actually a little bit of reactive oxygen species can play an important role in telling your body we've got low amounts of oxygen. So I know it's a little confusing because, wait, we've got low amounts of oxygen, but we're generating reactive oxygen species. And that's because we don't have enough oxygen to keep this pathway moving really fast. And so we get these holdups. So then the oxygen that we do have, because it's not being reduced in this normal way, now it's able to react with those free radicals that are being that are leaking out further up the line. Normally, however, the oxygen is going to be accepting the electrons. This is going to be used to pump out the protons. And all of this is going to be kept driven along at a nice pace because, well, then those protons are going to be flowing back through. You can think about it basically, if you don't keep taking protons in, you're basically creating a bigger gradient. And when you create a bigger gradient, yeah, you would get more power if it flows back in, but it's going to be harder to pump things out. And so ultimately, you're not going to have enough energy in order to keep protons pumping out if you're not able to do something and let those protons back in. We're not just going to let them in just directly. Instead, we're going to use the energy from letting them in to power the production of ATP. Because remember, when we've got a gradient, we move things down a gradient, thermodynamic favorability, we're going to release energy. We want to release that energy in a controlled way that's going to allow us to use that energy to make ATP. Turns out that there are things called uncoupling agents. That's basically, when we talk about coupling, we have this coupling of the production of ATP to the movement of electrons through this respiratory chain, through this electron transport chain. We can talk about uncoupling as if we basically uncouple the two. We make it so that the movement of electrons isn't going to be directly linked to the production of ATP. This could be a big issue or it could be helpful. So why might it be helpful? Well, it might be helpful if you need to generate heat, because basically that's what's gonna happen if you don't have them coupled. That energy's got to do something. When we talk about energy, we talk about like thermal movement and things, heat. And so if we were to uncouple the two, well, now we get the generation of heat, and maybe you're cold and you need some heat. Turns out that our bodies have this thing called brown fat. Adults don't have that much of it, but babies, like newborn babies, have a lot of it. And this brown fat, it's got a lot of this uncoupling protein. This uncoupling protein, what it does is it offers an alternative route for those protons to come back in, therefore dissipating the gradient. In the process, it's letting off heat because the movement of protons down the gradient is going to be energetically favorable. And as long as you kind of keep pumping protons out to keep creating the gradient and then just letting them flow back in, well, now you're going to generate heat. And this is going to allow newborn infants to stay warm enough. So good in some circumstances, but you can imagine that it would be bad and if it was out of control, because you wouldn't be using the energy to do ATP making. You wouldn't be able to make ATP. Instead, you just be making heat. This is what happens with uncoupling agents, things like 2,4-dinitrophenol, DNP, or this FCCP. This was actually used as like a weight loss drug, but they discovered that it was killing people because this uncoupling agent, it makes it so that you're able to burn fuel without making ATP, which could then be used to do things like build fat, but it's also going to make it so that you can't make the energy that your body needs. So do not take DNP as a weight loss tool. Instead, let's talk about using that energy to do something more useful. Make ATP, which is good. We need ATP. And so yes, if you have too much ATP because you take in too much fuel and then have a lot of ATP and then use that ATP to build fats, yes, that will make you fatter, but it's better than being dead because you can't make any ATP. So let's talk about how we make ATP. This is this really, really, really awesome molecule, this really awesome protein complex, this ATP synthase. So this is complex 5. This is also called ATP synthase. Sometimes you might see it called like ATPase. It's kind of confusing because it's going to make ATP, but it can also break down ATP because remember, an enzyme is going to catalyze things in both directions. And it turns out that if you don't have this proton gradient powering things, well, then what you'll actually do is you'll see that you can get things moving in the opposite direction. But we're going to be talking mostly about the forward direction when we're making ATP from ADP, an inorganic phosphate. It's this totally, totally beautiful molecule. And so I want you to go and take a look at the video. You might want to then rewatch the video after we go over the details to kind of let things sink in and contextualize things. But it's really this amazing, amazing machine. It's moving at things like 600 times per second or crazy speeds like that. We're really pumping out this ATP and it's so beautiful to see. So go watch it. So as you saw in the video, you have these two distinct multi-subunit components. We have our FO or F0, this integral membrane protein, and we have this F1, which is going to be our peripheral membrane protein. Our F1, this is where we're actually going to have the active sites in these beta subunits. And it's going to have this central stalk, this gamma subunit, that's going to connect things to our FO. Our FO is going to contain this A subunit, which is where the protons are going to flow through, as well as this ring of C units, the C ring. The C ring is going to turn when the protons flow through A. This is going to then be coupled because this is connected, we have the B is then also going to be connected to our F1. Basically, when you have this ring turn, it's going to cause this central stalk to turn. When the central stock turns, it's going to then lead to the kicking of the conformational changes happening in these F1 active sites. This is going to lead to the production of ATP. Well, actually, what it's leading to is kind of the release of ATP. One of the really fascinating things about ATP synthase is that the hardest step for it is actually releasing the ATP, so releasing the product. Normally, when we think about enzymes, we think about product release as being the fast step, that the hardest part was going and turning our substrate into our product. But then once we made that product, we'll just let go. Turns out that letting go is really hard for ATPase, this ATP synthase, because it binds to ATP really, really tightly. Binding to ATP really, really tightly, well, it enables it so that it's easier to make ATP, because you're getting basically the binding energy from binding to the ATP is going to make it so you'll have a lower transition state. This is going to make it so it's not as costly to make ATP as it would be normally. And in fact, you can actually go back and forth between ATP and ADP in the active site. But because it holds on to ATP so tightly, well, now it's going to be hard to let it go. How can you let it go? Well, you can let it go by changing the shape of that active site so that it doesn't have as high of affinity. In order to kind of coordinate that change, this is going to take place by having the movement of our FO, by the movement of the C ring, caused by the movement of protons, to cause the movement of the central stock, which is then going to kind of give a kick to the active sites that are holding onto the ATP. There's this idea, this theory, rotational catalysis, that is the name that we kind of give to this. In a little more detail, basically we had that F1 has three alpha and beta, three each of its alpha and beta subunits. The beta subunit, this is where we have our active site. The beta subunit can be in one of three conformations. The one that is, there's the one that holds on really tightly to ATP, this tight binding. Then we have the form and where it's like loose binding, it binds to ADP. And then we have empty, where it's very loose binding. So what we want to do is we're going to basically take the form, the conformation, the tight binding form, and we want to change it to the very loose binding form. And we can do this by kind of giving it a nudge. And we'll talk in a second about how we can give it a nudge. But when we give it a nudge, well, then we're not only nudging the one that we directly kick out that ATP, we're also going to cause a coordinated change through a cooperativity effect, where we're basically making the other subunits change their shape as well. Turns out that each of these subunits they have to be in one form, or like only one subunit can be in each of these forms. So they're all going to be in different forms, and they're going to take turns who's getting to make ATP. As we'll see in a minute, it turns out that it takes 3 protons in order to do one of those, to make one of those conformational changes. So it's going to take 3 protons to produce an ATP. And you would have to go through a complete rotation of this cycle would then synthesize 3 ATP, because you had those 3 sites. But for each site, it would take 3 protons per ATP. And so why do we get this number that it takes 4 protons per ATP? That's because you have to spend a 4th proton in order to bring in another phosphate, another inorganic phosphate. In order to keep the cycle going, we need to bring in more inorganic phosphate, bring in more ADP, and in order to make the ATP that we made useful, we need to ship it out. This will also help keep our product levels low and our reactant levels high. As you can imagine, these negatively charged molecules aren't just going to flow through the membrane on their own. Instead, they're going to need help. So they're going to need to travel through membrane proteins. In the case of our ADP and our ATP, turns out we're going to swap them through the same transporter. So this would be an example of an antiporter. because they're moving in opposite directions. Now, why are they moving in these directions? Well, we see that what we're doing is we're pumping out ATP4 minus and we're bringing in ADP3 minus. Based on charge alone, if we think about, remember, it's an electrochemical gradient, the electro part, we have things more positive on the intermembrane space, and therefore, and more negative on the matrix side. Therefore, if we pump out more negative charge than we bring in, from a charge standpoint, that's going to be favorable. And in fact, this is going to allow us then to do this swap. However, remember that the only reason that it was favorable was because we'd spent energy in order to pump out the protons. So although we're not directly seeing the protons involved here, that energy from the proton pumping was contributing. And so we're not changing proton concentrations on either side, but we are in effect dissipating the gradient of charge. So this is kind of costing us energy, but it's in an indirect way, and it's even more indirect than we normally think about when we think about our secondary active transport. Remember that our secondary active transport, we're driving something along against its electrochemical gradient, driven by an ion moving down its gradient. Well, if we think about if we think about getting the phosphate in, well, here we're actually going to use that kind of more of the classical secondary active transport. In this case, we're actually going to take a proton from the intermembrane side where it's got this high concentration of protons because we've been pumping them all out, and it's going to co-transport that with our inorganic phosphate. They're both coming in, so this is going to be a symporter because they're going in the same direction. They're both coming in, and we're paying the cost of this inorganic phosphate coming in by using the gradient that we created. Because we're directly using this proton here, then we cost an extra proton per ATP that we produce. So we need those 3 to turn the pump, and then we need the 4th to facilitate the co-transport of bringing in this inorganic phosphate to keep things going. And then indirectly, that the gradient we're producing is also being used to power this other swap. But when we think about direct numbers for our accounting purposes, we typically just consider that there's going to be this one extra proton that you need, because that's the one that's going to be directly related to the transport of that inorganic phosphate. Whereas here, it's just like an indirect effect, and we're not directly transporting a proton, we're just changing the relative charges on the sides and lowering the electrochemical gradient that way. The electrical part, but not the chemical part. How are we going to, however, connect the movement of protons to the actual making of ATP? We connected it to the flow of the proton, of the inorganic phosphate, but really what we care about is how is the actual movement of protons going to be able to cause the making of ATP? It turns out that this is actually pretty cool. So in that A subunit of FO, so this part, remember FO, it's this integral membrane protein, so it's embedded in here. We have this A part. It kind of has these 2 little channels where one is open, these half channels. So one of these half channels is open to so in this case, again, it's upside down. So right here where we have a lot of protons, where is that going to be? Right, that's going to be in our inner membrane space. And then up here where we don't have as many protons, what's that called? Yeah, that's going to be our matrix. So we have the side that's open to the matrix and the side that's open to the intermembrane space. And each of these is kind of going to be, is going to come into contact with the C ring. So the C ring, we've got this, they have different species have variable numbers of these C subunits. What's going to happen is each of these C subunits has a binding site for protons. This binding site is either going to be a glutamate or an aspartate, depending on the species. So remember that each of these can basically give and take protons. We typically think about them as having given up protons, but you can imagine that if there's a lot of protons around, then they're going to be protonated. And where would you find a lot of protons around? Well, you find a lot of protons around in the inner membrane space. So when that glutamate or when that aspartate is exposed to the concentration of protons, so to the pH that's present in the inner membrane space, well, now it's actually going to protonate. But on the other side, when it's exposed to the matrix, where there's a low concentration of protons, well, there it's going to deprotonate. So if we can expose that critical residue to one side and then change and expose it to the other side, we can then transfer a proton from one side to the next. And in fact, that's what happens. So you have protons flow in. It can then protonate that key residue. That's going to interrupt interactions that residue had to another residue. And so basically, you're going to interrupt ionic interactions. When you interrupt things, then you kind of are going to cause a conformational change. This is going to drive the movement of this ring. So you get protonation. It's going to cause a conformational change. The conformational change is going to move the position of that key residue into the half channel where it can actually sense the environment in the matrix. When it senses the environment in the matrix, it's going to release the proton. In the process, you had this conformational change occurring. You had this movement of the ring, and the ring wasn't isolated. Instead, it was attached to these other parts of the protein. And so when this rotates, you get this rotation of the gamma subunit. And when the gamma subunit rotates, well, now it's going to cause conformational changes in the beta subunits of our F1. And those conformational changes in our F1, well, that's how we were going to be kicking out those phosphate, the ATP. So ultimately, we get this idea of rotational catalysis, where we have a rotation of the C ring linked to the production of ATP. Now let's do a little bit of ATP accounting. So imagine that we have NADH. It's going to come in complex 1. In complex 1, we're going to pump out 4 protons. And we're going to be passing a pair of electrons onto Q. That pair of electrons from Q is then going to go and it's going to travel to complex 3, and it's going to pass those electrons onto cytochrome C. In the process, it's going to pump out 4 protons. So we get 8 protons so far. That cytochrome C is now going to go and it's going to take those electrons to oxygen in complex 4. In doing so, it's going to pump out 2 protons. So ultimately, from our NADH, we got 10 protons pumped out. Now, we said that it took 4 protons to make an ATP because we had 3 to directly make it and then 1 to bring in the inorganic phosphate. So how many ATPs would we get from an NADH if that NADH was made in our matrix? Well, 10 divided by 4, we've got 2.5. But in the case of our FADH2, so remember that we're going to be going in complex 2. There we don't get those original 4 protons because we're not going through complex 1. So instead we get 6. We get 6 divided by 4 is 1.5. So we get about 1.5 ATP per FADH2. This ratio is something called this P to O ratio or this P to 2E minus ratio, 2 electron ratio. And so that's where we get this about 2.5 and about 1. And it's okay that we're getting these ratios and we're not really sure about numbers and things because remember, it's the gradient that's going to be doing the work. And it's not like there's a direct correlation between a number of NADH and the number of ATP. Instead, the NADH is contributing to this gradient. The FADH2 is contributing to this gradient. And ultimately, this gradient is going to be what's driving things. But it doesn't need to be like there's a distinct numerical ratio that makes total sense. With those caveats in mind, let's go and let's think through what we would get from breaking down glucose, both in terms directly of ATP as well as our NADH, our FADH2, and then how much we'd end up with in the end. Going stepwise, we take that glucose and we're going to oxidize it through glycolysis to give us pyruvate. In the process, remember, we spent 2 ATP, but then we made 4 ATP, giving us a net of 2 ATP. We also generated 2 NADH, and so those 2 NADH, well, now they can go and they can contribute later down the line to our electron transport chain and our oxidative phosphorylation. I also want to point out that, remember, we saw an example of oxidative phosphorylation when we talked about glycolysis. So this step from glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, this was actually going to be an example of oxidative phosphorylation because we were both oxidizing and phosphorylating. But in this case, these were kind of coupled together. Whereas in the oxidative phosphorylation we talk about in terms of our ATP generation, there we have a decoupling of the oxidation and the phosphorylation. So they're not directly linked. Instead, the oxidation is used to make the gradient, and the gradient is used to drive the phosphorylation. So they don't need to be directly linked. But here, the energy from the oxidation is used to directly add on the phosphate. In the case of the oxidative phosphorylation we typically think about, there, the energy from the oxidations is used to create the gradient, and the gradient is used to drive the phosphorylation step. That in mind, let's go back to our story about glycolysis and our story of the lifetime of this glucose. From the glycolysis, we got this 2 NADH and 2 ATP. Then we went and we said, okay, well, we want to get the bigger payout. We want to keep oxidizing our pyruvate. In order to keep oxidizing our pyruvate, we're going to need to get it into the citric acid cycle. But our citric acid cycle, well, to get into it, we need to be in the form of acetyl-CoA. And so that's why we had the oxidative decarboxylation step of pyruvate. Basically, we kicked off that CO2, and instead we linked it up to a CoA molecule to give us acetyl-CoA, which could then enter the citric acid cycle. Because the name suggests oxidative decarboxylation, we were having an oxidation reaction, which means that we were reducing something else. In this case, we were reducing NAD+, and so we generated NADH. We had two pyruvate molecules, so we're going to generate 2 NADH. And in this case, what we're doing is we're actually doing it in the mitochondrial matrix, and this is going to be a key factor in a minute. Because remember that we need to be in the matrix in order to enter complex 1. We need to actually be in this matrix. When we're generating the NADH in the glycolysis steps, that was going to be happening out in the cytoplasm, not in the matrix. And so we're going to have to go through some tricks in order to get that in. So keep that in mind. But going back to our story, we went through pyruvate oxidation and we got our 2 NADH. Now let's go and let's take that through the citric acid cycle. When we do this, well, now remember, we had 2 pyruvates, so we get 2 acetyl-CoAs, and so we're going to do this cycle twice. And so on net, we're going to get 6 NADH, 2 FADH2, and remember that that's our complex 2. This step is actually happening, our complex 2 step, and we're going to get the 2 GTP per turn. And then ultimately, those are going to then go into our electron transport chain. We said that if we're entering at complex 1, if our NADH is entering at complex 1, we're going to get 2.5 ATP per NADH. If we entered, we had FADH2, which we have in our complex 2, which we had in our, that's our succinate dehydrogenase step. Well, there we're only going to get 1.5 ATP. So ultimately, on the high side, we would have 32 ATP. But you'll see that when it says like final ATP, it says like 3 or 5 in the case of glycolysis. Well, that's because this NADH that you made was going to be made in the cytosol. And remember that the cytosol, well, that's not where we need to have, that's not the side where complex 1 is going to start. If we want NADH to enter complex 1, we need to get it into the mitochondrial matrix. And this membrane is going to be very picky, and it's not going to let in NADH. So cytosolic NADH can't get into the mitochondrial matrix. So instead, we have to use shuttles to get reducing equivalents in instead. There are two main shuttles, the glycerol 3-phosphate shuttle and the malate aspartate shuttle. The brain and muscles use this glycerol 3-phosphate shuttle in order to get these NADH equivalents in. And they're going to do so by passing electrons onto Q. How it works is it involves dihydroxyacetone phosphate, so our DHAP. You might remember this as one of the things in glycolysis. And in glycolysis, well, we went and we used our triose phosphate isomerase to isomerize that into glyceraldehyde 3-phosphate, and that's what we cared about. Note that this was glyceraldehyde 3-phosphate. Here we're talking about glycerol 3-phosphate. So here we're actually reducing this ketone to an alcohol. In doing so, what we're going to do is we're actually going to be using the NADH from glycolysis in order to do so. So we're using the NADH from glycolysis to then transfer those electrons to this dihydroxyacetone phosphate to give us glycerol 3-phosphate, not glyceraldehyde 3-phosphate. This glycerol 3-phosphate, well, now it can go and it can transfer those electrons onto FAD to give us FADH2 and recreate dihydroxyacetone phosphate. So we reduce dihydroxyacetone phosphate with our NADH, and then that gave us glycerol 3-phosphate. We then oxidize that with FAD to give us back our dihydroxyacetone phosphate and our FADH2. Remember FADH2, well, that could pass electrons onto Q, so that could enter complex 3, but that's not going to be able to go through complex 1. If we don't go through complex 1, well, now we're not going to get the pumping of electrons. This would be only give us 1.5 ATP per NADH because that we made out in this and the cytosol. So we're losing some of our energy. Turns out there is a way to conserve that energy. The heart, liver, and kidneys use this thing called the malate aspartate shuttle to get NADH equivalents into the electron transport chain. They're still not going to be able to transport NADH into the matrix, but they're going to directly produce NADH in the matrix using electrons from NADH that was in the cytosol. It's a complicated kind of diagram and thing to look at, and don't worry about all the details, but it involves this idea about transamination. We'll see this when we talk about amino acid metabolism, but basically you can transfer the amino group from a free amino acid onto alpha ketoglutarate to give you glutamate, as well as an alpha keto acid. So we can do this, say, take this aspartate amino transferase, transfer an amino group from aspartate onto alpha ketoglutarate to give us glutamate and oxaloacetate. Turns out we can take that oxaloacetate and we can reduce it to form malate using malate dehydrogenase. Malate, it can go through the membrane, the inner membrane. Oxaloacetate couldn't, but malate can. When malate goes in, it goes in through this inner membrane, this malate alpha ketoglutarate antiporter. So it goes in, then it's going to, we have malate dehydrogenase in this side of the membrane. So in our matrix is going to then, it's going to oxidize it back. When it oxidizes it back, it's going to pass those electrons onto NAD plus to give us NADH. And that NADH can then go and it can enter complex 1. So we get the whole payout. The rest of this complicated stuff is just to regenerate things. So we have to basically reverse everything that we did on the cytoplasmic side. We have to reverse it in the matrix side in order to keep things going. And then we just have a transporter that can swap things back. So we have our aspartate amino transferase. It's going to take that oxaloacetate that we just made, and it's going to take glutamate and it's going to do that swappy-doppy to give us alpha-ketoglutarate and aspartate. The aspartate can then leave, and in return, it's going to bring in another glutamate so that you can keep this cycle going. The bottom line is that we take those electrons from our NADH, we transfer them onto another molecule that's able to then enter the mitochondrial matrix. So we take oxalic acetate and we reduce it to malate. That malate can come in. When it comes in, we can then reverse the process. So we take those electrons off of malate and put them back on NAD plus to give us NADH, and that's then able to enter the electron transport chain. So we took this trick in order to get it so that we can enter at the complex 1. And if we do that, well, then we get our full yield. Big picture, all of this is happening kind of at the end of the line. Not exactly sure why I have 26 to 28 ATP here, but the numbers you see vary widely. And because it depends on things like the number of C subunits, it depends on things like if you're using the gradients, leakiness, all of this various stuff. So it's you'll see a variation in numbers, because remember that you don't have a direct 1-to-1 correlation or even at 0.5 to 1 correlation or anything like this, because really what the NADH is powering, what the FADH2 is powering, they're powering the creation of the gradient. They're not directly powering the production of ATP. And in fact, we can use the gradient for other things, as we saw for transferring in a phosphate group. You can do things like power the import of calcium, all sorts of stuff. And all of this was kind of happening at the end of a bunch of interconnected pathways. If we think about, we talked a lot about how these other pathways, things like glycolysis and the citric acid cycle, were going to be regulated allosterically by the availability of ADP and ATP and NADH, as well as thermodynamically by them. With oxidative phosphorylation, here what we're really regulated by is going to be, our availability of our ADP and oxygen. If we don't have a lot of ADP, well, that would indicate that we don't need to keep making ATP, because if we don't have a lot of ADP, we probably have a lot of ATP. So we're not burning energy, and so we don't need to keep making ATP. And we can instead do things like use the ATP that we have. And if we don't have enough ADP, well, that is going to slow things down, because we don't have enough of our reactant to keep driving things. And we talked about earlier how if we don't have enough oxygen, we don't have enough of our acceptor, our terminal acceptor. Well, now our chain is going to get backed up. And if our chain gets backed up, then we can get things like reactive oxygen species. A little bit of those can be good, because they can signal to our body like, hey, we don't have enough oxygen, but too much is going to be bad. There are also other regulatory methods, but bottom line is all of these are going to be interconnected, and the availability of oxygen and the availability of ADP are ultimately going to be what is going to dictate that ox-phos can happen. And then assuming we have that tight coupling, we're going to make ATP from the power of the electron transport chain. So remember that we go in through complex 1, or we go in through complex 2, or any other way to Q. If we go in through complex 1, though, then we're going to get more pump. We're going to get to pump out protons. Regardless, once we get to the electrons to Q, no matter how we get them through Q, now they're able to go to complex 3. Complex 3 passes those electrons onto cytochrome C. Cytochrome C is then going to go, and it's going to pass those electrons onto oxygen in complex 4. Both complex 3 and complex 4, we're going to pump protons out along the way, and them together with the protons pumped out in complex 1, this contributes to creating a proton gradient. When you have this large separation of both charge as well as kind of chemical species, we get this large electrochemical gradient that provides the proton motive force that's going to allow it to flow through like a hydrolytic dam-like mechanism-y thing, where the flow of electrons is going to be coupled to the movement of this ATP synthase. The movement of the ATP synthase is then going to make it so that you produce ATP. It costs about 4 protons per ATP made. And so if you start with an NADH, you're going to get about 2.5 protons. If you start with an FADH2, you're going to get about 1.5 protons. And then we just need to consider where the NADH was coming from, if it was already in the matrix or whether it was in the cytoplasm. When trying to do some sort of like accounting for how much you get. But those numbers are all kind of just estimates and things. And the key thing is really just this concept that you're passing electrons on from one thing with a lower reduction potential to something with a higher standard reduction potential, using the energy from those transfers to power the pumping of protons, which powers the production of ATP.